Now we are going to understand the heterotrophic mode of nutrition in the following slides. It is a wide range of nutritional mode. In heterotrophic mode of nutrition, organisms depend on some other organism for the requirement of their food. We can understand heterotrophic mode of nutrition by some examples, like example of fungi and bacteria. They are having extracellular digestion process by which they are secreting the enzymes to the substrates outside the body, and once the food is digested they absorb them. But in case of the human being, the process of digestion takes place inside, so enzymes are being released in the digestive system, and thereafter it is being absorbed by the body cells. Not only the animals which fall under the category of heterotrophic mode of nutrition, there are certain exceptional plants like Cuscuta which comes under the category of heterotrophic mode of nutrition. Cuscuta plant is devoid of root, stem and leaf. Therefore, they are growing on some host plant and they derive the nutrition from them directly. Nutrition in animals. Animals in general having a wide range of modes by which they are taking their foods. Some are herbivores, some are carnivores, some are very small parasites. So we see a lot of diversity is there in their mode of nutrition, in the heterotrophic mode of their nutrition. But one thing remains common in all of them, that they take up the complex organic material from the nature or some other organism and simplify them so that it could be utilized by their cell. So, simplifying the complex organic material by the enzymatic process, enzyme-based simplification, that is digestion process, is known as holozoic mode of nutrition that we find common from amoeba to the human beings. We will try to understand the nutritional mode in amoeba, which is the holozoic one. Amoeba is unicellular organism, so whenever any food comes in contact with the cell membrane of the amoeba, it sends out its membrane toward the food so that it could encircle it from all sides. That newly formed structure which traps the food from all around is known as pseudopodia. So the mechanism of trapping food is simple. It traps it from the all side so that it could be inside a membrane-like structure which is called as food vacuole. Now once the food vacuole is been formed, amoeba releases enzymes so that the food trapped inside the food vacuole could be digested. Now under the influence of the enzyme, the food is been digested, and once the process of the digestion is completed, the simplified food comes into the cytoplasm of the amoeba. Now, whatever is left inside the food vacuole is of no use for the amoeba. Therefore, that waste vacuole moves toward the membrane. Once it gets attached with the membrane, the adjoining areas dissolves and the waste is being released out. So we find even in case of the amoeba, there are following process of digestion is taking place. First, ingestion, when the food is being trapped inside the food vacuole. Second, digestion. However, amoeba do not have the digestive system, but here we find the food vacuole of amoeba is analogous to the digestive system. After the digestion, the absorption and assimilation process is simultaneous, and finally the waste containing vacuoles comes out to release the waste in them by the process called as egestion. So therefore we find the amoeba is very common in the process of digestion with that of the human beings. The process of taking food in amoeba is known as phagocytosis process. Let us try to understand the nutritional mode in human beings. As we have discussed earlier, amoeba and human beings both are having the holozoic mode of nutrition, but there is a range of differences between them, like human beings are having well-defined digestive system which was not present in case of amoeba, and human beings are able to digest more complex food like carbohydrate, fat and protein that could not be digested by the amoeba. Human digestive system having two major parts. One is the alimentary canal and other is the digestive glands. Alimentary canal is been defined as long tubular structure that starts with the mouth and ends at anus. Alimentary canal is not uniform in its structure along its length. The other parts of the alimentary canal are esophagus, stomach, small intestine, and large intestine. So, one by one, we will try to understand the parts of the alimentary canal. First is the buccal cavity, which is also called as oral cavity. It is the space inside the mouth where teeth and tongues are present, and it has been utilized for chewing, cutting, and masticating the food. The buccal cavity of our mouth also receives enzymes from our salivary gland that help in the process of digestion of starch. So first food is being chewed and masticated with the help of the enzymes, and the food in the mouth is called as bolus. Once the food is being prepared as a bolus, it goes to the stomach by a long tube which is called as esophagus. However, the process of movement of food from esophagus to the stomach is being accompanied by the longitudinal and circular muscles or the esophageal wall. The movement of these muscular structures is known as peristalsis. Digestive glands are the exocrine glands. They release various kinds of enzymes at the different stages of digestion through the length of alimentary canal. They help in the digestion of various types of chemical compound which are commonly very complex in nature. One by one we will try to understand the different type of digestive glands and the type of enzymes they are releasing. First in order is salivary gland, which is found in our mouth, and it releases an enzyme which is known as salivary amylase. Salivary amylase digests the starch into maltose. Second is gastric gland, which is found in our stomach. It releases mixture of enzymes like pepsin, gastric lipase, and amylase. Pepsin is a proteolytic enzyme. It digests the protein, whereas gastric lipase digests the fat and amylase digests the starch. However, the environment for the operation of gastric juice should be the acidic. So, the one of the component of the gastric juice is the hydrochloric acid and mucus. Hydrochloric acid provides the environment for the working of pepsin enzyme, and also it helps in killing the germs. Mucus further help in the protection of the gastric wall, and it also helps in the process of movement of food. Third is the liver. It is the largest endocrine gland, release green liquid known as bile, which contains certain pigments. However, the bile is not being stored in the liver, rather it is being stored temporarily in a sac-like structure called as gallbladder. The function of bile juice is to help the process of digestion of fat. It helps the digestion of fat by a process called as emulsification. In that process, the large fat molecules are being broken into small fat droplets so that the lipase enzymes can work them to digest properly. Pancreas is a hypocrine gland. It is partially endocrine and partially exocrine. Its exocrinal part releases trypsin, pancreatic lipase, and amylase. Trypsin is again a proteolytic enzyme and it works in an alkaline medium, whereas pancreatic lipase help in the process of digestion of fat and amylase help in the process of digestion of starch. Gallbladder is a green sac-like structure. It stores temporarily the bile in them. Now we will try to understand the process of digestion in stomach in a bit detail. As we have seen in the previous slide, that the stomach receives a mixture of enzymes called as gastric juices, which contains pepsin, gastric lipase, and gastric amylase. It also releases hydrochloric acid and mucus. Pepsin requires acidic environment for their proper action, whereas mucus protects the lining of the stomach. After the digestion of food which takes place in the stomach has been completed, it moves to the small intestine. The small intestine is the largest part of our alimentary canal. It is further divided into duodenum, jejunum, and ileum. Among them, duodenum is the most important part where it receives secretion from pancreas. So, pancreatic juice, as we have seen, that contains trypsin, pancreatic lipase, and pancreatic amylase. However, due to the effect of bile, because bile is also being released in duodenum through a common duct called as hepatopancreatic duct, makes the environment of the food totally alkaline. And trypsin is the enzyme which requires alkaline medium for their proper action. So again we see the process of digestion of protein remains continued. It breaks up into peptides and amino acids. Fats are being digested more properly, as we see that under the influence of bile, the large fat molecules are being broken into small fat droplets so that the lipase enzymes can digest them properly. Similarly, the carbohydrates are further digested into the simple sugars. Once the food is digested into the duodenum, it passes to the next part called as jejunum. Here again, some other enzymes, chemotrypsins and lipases, works on undigested foods and digest them properly. But it is the last part of the alimentary canal where digestion of the food occurs. Once the food moves to the ileum, no digestion process is taking place. The structure of the ileum wall is very different than rest of the other part of the small intestine. In the figure we can see the inner wall having many finger-like projections called as villi. These villi are formed in that way because they have to increase the surface area for the absorption of food. Underlying their wall, there are network of capillaries of blood vessels and lymphatic duct for the absorption of digested food so that the proper digestion of so that the absorption of the food should take place. Now, ileum is the area where absorption of food is taking place. Whatever been digested up till now will be absorbed from here and all the simplified food will go to the blood. This process is known as absorption. Following the absorption is the assimilation. Now, each and every cell of the body receives their kind of nutrients from the blood. So, when the nutrients are being absorbed by the cells and tissue from the blood, the process is called as assimilation. So, we have seen that the process of digestion starts in the mouth and the first digestion of the nutrient takes place in the mouth. It is carbohydrate which has been digested first, but later on the food when it comes into the stomach, next nutrients like fat and proteins are also being digested. However, we see the proper digestion of the fat is only taking place in the small intestine. However, all the parts of the small intestine is not helping in the process of digestion. We see in case of the ileum, it only helps in the absorption of the food. So whatever is being digested up till the ileum part will be absorbed and assimilated in cells and tissue. And the rest of the food which could not be digested will move to the large intestine. Large intestine has many folds inside to increase the surface area for absorption, called as colons. There is no digestion in large intestine, only absorption of water, minerals, drugs, pigments and vitamins are taking place there. The undigested food further moves to the rectum and disposed of through the anus. Now the process is called egestion. However, the disposal of food, undigested part of the food, is being regulated by a sphincter known as anal sphincter. We have seen the mode of nutrition in animals are usually holozoic, where the complex food are being taken and they are acted upon by different kinds of enzymes so that the complex food may be digested into the simple one and they are being absorbed and assimilated in cells. We have found a degree of similarity in case of amoeba and human beings so far the process of digestion is concerned. However, amoeba is a unicellular organism and human beings are the complex organism. They vary in terms of the food they are taking. Amoeba is taking simple food whereas human beings are taking variety of food items and they are very complex in nature. In order to digest these complex substances, human beings has been evolved into a very complex system of enzyme that is called digestive glands. Various types of digestive glands are very specific in terms of their secretions. They are containing a wide range of enzymes to digest the various kinds of complex substances. We have also seen in the process of digestion, no digestion is taking place in esophagus, ileum, and large intestine. Large intestine is a very important area because it is the place where absorption of water, minerals is taking place. Dental caries or tooth decay is a gradual softening of our enamel and dentine. It begins when bacteria acting on sugar produces acids to demineralize the enamel. Masses of bacterial cells along with organic food particles stick to the teeth to form dental plaques. Saliva, due to these deposition, cannot reach to the tooth surfaces to neutralize the acid. Therefore, the process of demineralization remains continued and finally it causes the formation of cavities, pulpitis, inflammation and all. So it is very important that one should maintain the good dental hygiene by brushing twice in a day. Thank you for watching the video.